Welcome back to Science Spotlight. Today, we are joined by our student expert to break down meiosis. Thanks for being with us today. Let's start simple. What is meiosis? Thank you for having me. Meiosis is a type of cell division that makes gametes, like sperm cells or egg cells. It takes one diploid cell and turns it into four unique haploid cells, and it happens in two rounds, meiosis 1 and meiosis 2. Great! Let's dive into meiosis 1. What happens first in prophase 1? Great question. In prophase 1, the chromosomes condense and homologous pairs match up. They also do something important called crossing over, where they swap little DNA segments. This creates genetic variation. So what happens in metaphase 1? In metaphase 1, the homologous pairs line up side by side in the middle of the cell. The way they line up is random. That's called independent assortment. Another reason we all look different. Then we move into anaphase 1, right? Yep. In anaphase 1, the homologous chromosomes separate and get pulled into opposite sides of the cell. Interesting. And how does meiosis 1 wrap up? Meiosis 1 wraps up in telophase 1. The cell splits in half. Now we have two cells, and each still has duplicated chromosomes, so we're not done yet. So let's jump into meiosis 2. What happens in prophase 2? In prophase 2, the chromosomes tighten back up and new spindle fibers form, kind of like a reset. Then metaphase 2. In metaphase 2, this time, chromosomes line up single file, in the middle, not in pairs. And anaphase 2? In anaphase 2, the sister chromatids finally split apart and move to opposite ends of the cell. And finally, the last step, telophase 2. What do we end with? And finally, telophase 2. The cell divides again and we end up with four haploid cells, each genetically unique. That's the whole point, variation and proper chromosome numbers for reproduction. Amazing breakdown. Thanks for joining us and walking us through meiosis.